Great, everybody. Well, I think people are still trickling in, but the trickle has slowed, so I'll get started. I want to welcome everyone to this month's installment of the ASBMB Lipid Research Division Seminar Series. Today we have two fantastic speakers lined up that work at the interface of lipids and organelle and mitochondrial biology and mitochondrial organization. We're going to learn about lipids and how the inside of mitochondria interplay with each other. And then in the second talk, we're going to hear about interorganelle contacts and how they influence both mitochondrial internal organization or overall organization, and then also PI4P distribution at the plasma membrane. So before I introduce the first speaker, I wanted to let you know that we'll take questions at the end of each talk. And rather than typing your question into the chat box, if you'll use the raise your hand function, I can call on you and then you'll have a chance to unmute yourself and ask your question. So for the first talk today, I want to introduce Kailash Venkatraman. He's a fifth-year graduate student in Itai Budin's lab at UC San Diego. He's going to tell us about his work on the interplay between the mitochondrial lipid cardiolipin, lipid saturation state, and mitochondrial ultrastructure. I also want to point out that he is going to be graduating pretty soon and is on the market for postdocs. So keep him in mind as a potential future postdoc in your lab. And so with that, I'll let you take it away, Kailash.
Thank you, Jonathan, for that very kind introduction. Let me just share my screen. I hope you can all see this. Okay, yeah. So, yeah, thank you, Jonathan. So I'm really excited to sort of tell you about a new story, sort of a couple of stories related to how lipid composition can influence the structure and function of mitochondria, with particular relevance for the pathology known as Barth syndrome. Okay. So I likely don't need to tell anyone within this audience, but mitochondria are the central hubs of bulk ATP production within our cells. They're consistent of an outer membrane, which encapsulates an invaginated inner membrane, whose inward folds have come to be known as the cristae. These cristae membranes are extremely high in curvature, and they house some of the crucial bioenergetic complexes required for cellular respiration, such as the electron transport chain. And given the sort of functional importance of cristae for cellular energy production, much research has been dedicated to understanding the molecular determinants of this uniquely invaginated topology. And indeed, over the past 20 years or so, this research has implicated three main proteins in shaping cristae structure, the best characterized of which is ATP synthase, which forms dimers and localizes to cristae tips, where it can bend the membrane locally and also self-assemble into these dimer-like rows in regions of positive curvature on cristae membranes. Analogously, at cristae junctions, MECOS and OPA1 can physically and functionally interact to tighten cristae junctions, stabilizing cristae structure and inducing negative curvature. While the functional and physical interplay of these proteins in shaping cristae has been very well defined in the literature, analogous roles for mitochondrial lipids in shaping cristae structure is an area of emerging study. Indeed, the mitochondrial lipidome itself is actually extremely unique. For one, it harbors a unique phospholipid known as cardiolipin, which is exclusively synthesized and localized in the inner mitochondrial membrane. And in contrast to bulk phospholipids like phosphatidylcholine, which have one phosphate in its head group and two acyl chains, giving rise to these cylindrical-like structures, cardiolipin has four acyl chains and two phosphates in its head group, and this difference in cross-sectional area between head group and acyl chain give rise to these inverted conical-like structures, which have a large negative intrinsic curvature. Now, dysregulation of cardiolipin's acyl chain composition has been implicated in causing the pathology known as Barth syndrome, where patients exhibit significant cardiomyopathy, muscle weakness, and susceptibility to infection. And a hallmark of the Barth syndrome pathology is that in patient cells, there's significant aberration to cristae structure. And it was sort of the seminal finding that cardiolipin could be implicated in shaping cristae structure, but the mechanisms that govern cardiolipin's role still remain mysterious. Another sort of unique aspect of the mitochondrial lipidome is its specific enrichment in unsaturated phospholipids, which in contrast to their saturated counterparts exhibit large negative curvatures. And indeed, in the mitochondrial field, it's long been shown that supplementing cells in cell culture with saturated fatty acids like palmitic acid have long been shown to induce mitochondrial dysfunction. However, the mechanisms governing this dysfunction, the causes of this dysfunction still remain mysterious. Indeed, we wanted to better understand how saturated fatty acid accumulation could cause mitochondrial dysfunction, and therefore we turned to a system within yeast whereby we could manipulate the levels of saturation through manipulation of the sole lipid desaturase within yeast, known as OLI1. We constructed 4 different strains with discrete levels of saturation and decreasing levels of OLI1 expression, entitled SFA1 through SFA4, saturated fatty acid 1 through 4. And indeed, we verified that these strains change lipid saturation via lipidomics, where we see a transition from predominantly diunsaturated phospholipids, which is the maximum level of unsaturation within yeast, which cannot metabolize PUFAs, polyunsaturated fats, to predominantly monounsaturated and saturated species in SFA4, a trend that was retained in each of the major phospholipid classes within yeast, PC, PE, and PI. Now, when handling these strains and observing their mitochondrial morphology by confocal microscopy, we observed that a mild increase in saturation, such as in SFA2, retained interconnected mitochondrial morphologies, but a small increase in saturation in SFA3 resulted in fragmented mitochondrial structures and eventually completely ablated mitochondrial structures, which correlated with a complete seizure in respiratory capacity. Indeed, when observing these highly saturated strains using electron microscopy for their cristae ultrastructure, we observed 2 distinct types of aberrant cristae structures, which mimicked defects or losses to cristae shaping proteins. For one, we observed these onion-like inner mitochondrial membranes with multiple inner membranes, which resembled a loss to ATP synthase dimerization induced by knockout of ATP20. And also these completely flat inner mitochondrial membrane structures, which were bereft of cristae completely and mimicked a deletion to the yeast ortholog of OPA1. Indeed, the hallmark of these structures were that they were completely or were largely bereft of membrane curvature, which was in stark contrast to the healthy cristae that I showed earlier. But we were still wondering what the sort of mechanism was that's driving these dysfunctions induced by high saturation. And indeed, we found that an alteration in ATP synthase organization as a function of saturation. ATP synthase predominantly exists as oligomers or dimers in highly unsaturated conditions, but increasing saturation caused a disassembly of ATP synthase dimers into predominantly monomers in highly saturated strains like SFA3 and 4, which mimicked the organization of ATP synthase in ATP20 knockouts, which suggested to us that this loss of dimerization could be driving these mitochondrial defects that we observed in SFA strains. Indeed, we saw some interesting changes in the mitochondrial lipidome itself as a function of saturation. So in the whole cell, we see a decrease to the PE to PC ratio, which is sort of indicative of an adaptation to membrane fluidity, as PC is more fluidizing than PE. However, in the mitochondria, we actually see the opposite effect. We see an increase in the PE to PC ratio, which is counter to this idea of adapting to membrane fluidity, but supports this idea that there's an increase in synthesis of curved lipids, like as PE is a higher intrinsic curvature than PC. But we were still wondering how these mitochondrial lipid-specific changes can affect membrane physical property. And to further elucidate this, we turned to molecular dynamics simulations in collaboration with Chris Lee, who is then a postdoc at Padmini Rangamani's lab, but now runs his own group at UCSD. And Chris conducted coarse-grained molecular dynamics simulations, which forego atomistic detail in favor of the ability to simulate complex systems. And indeed, what Chris observed through stimulating the lipidomes, realistic lipidomes from our SFA strains, was that while there was an increase in the stiffness of the membrane itself in these lipidomes, there was a retention of curvature as increasing saturation occurred. And he specifically predicted that cardiolipin was instrumental in this process of retaining membrane curvature as a function of saturation. So we were wondering, is there a specific interplay between cardiolipin and increasing saturation that could be shaping cristae structure? And to do this, we knocked out cardiolipin synthase within our SFA strains to see any epistasis. And what we saw is that a loss of cardiolipin in unsaturated growth conditions retained cristae tubules and interconnected mitochondrial morphology, which has long been sort of a mystery within the field and has been observed before, given cardiolipin's multitude of functions in mammalian cells. It's a bit surprising. But then a mild increase in saturation retains interconnected mitochondria with a retention of cristae tubule formation. But a mild increase in saturation and loss, coupled with a loss of cardiolipin, resulted in ablation of mitochondrial morphology and the formation of onion-like cristae structures, which occurred despite the retention of ATP synthase dimers, suggesting that cardiolipin could be shaping cristae sort of independently of ATP synthase dimerization or the cristae shaping machinery itself. And to sort of further understand this, we integrated our results into a mechanical model, which synergized the curvature contributions of cardiolipin and the curvature contributions of ATP synthase dimers in shaping cristae tubule formation and lengthening. And what we see in this mechanical model, which was done in collaboration with Padmini Rangamani at UCSD in mechanical engineering, is rather than a gradual increase in cristae tubule length and formation, we see a critical mechanical threshold which drives the formation of these characteristic cristae tubules, which is often termed as a snap-through instability between these two cristae states, much like these snap bracelets, which I think I'm a bit too young to have ever actually seen, but I've heard that were an absolute fad within the 90s. So this transition is much like these toys in the 90s. And indeed, we sort of observed that the sort of interplay between both cardiolipin and unsaturated phospholipids mediate this snap-through instability from flat inner mitochondrial membranes to characteristic cristae structures. But we wanted to apply this understanding and this model to further understand how natural changes in the lipidome could affect the role and functionality of cardiolipin. And indeed, we thought about how we grow yeast in the lab. And indeed, yeast in nature grow predominantly on plants or on fruits in significantly lower oxygenated environments compared to the way we grow yeast in lab in 200 RPM baffled shake flasks. And this is important for shaping the cellular lipidome because OLE1, the lipid desaturase within yeast, is an oxygen-dependent enzyme, and therefore limiting oxygen, such as in the natural yeast environments, could result in changes to lipid saturation. And indeed, when we grow cells microaerobically and limit oxygenation, we see an increase in lipid saturation, which sort of mimics actually the double bond profile of mammalian cells, which are more saturated than the artificially unsaturated yeast. Indeed, we see a surprising increase in the levels of cardiolipin, which is sort of counter to its traditional role in aerobic metabolism. And further, when we grew cardiolipin deficient cells under microaerobic conditions and looked at their mitochondrial structure and function, we saw that its loss completely ablates mitochondrial morphology and cristae ultrastructure, suggesting an essential role for cardiolipin synthesis in maintaining mitochondrial structure in these conditions. And so we see an essential role for cardiolipin synthesis, specifically under these saturated conditions. And we wanted to further apply this to understanding a process known as cardiolipin remodeling, which is effectively the way the mechanism by which unsaturated fatty acids are specified to cardiolipin. And indeed, this process is conserved from yeast to mammals and is particularly important for the pathology I described earlier, known as Barth syndrome, which is caused by mutations in tafazin, which is the human ortholog of TAS1P. And indeed, much of the seminal findings regarding the mechanisms of cardiolipin remodeling and its roles were initially elucidated within yeast. But it's been sort of complexified by the fact that many studies have observed a lack of sort of functional difference between unremodeled and remodeled cardiolipin within yeast. And we posited that perhaps this could be explained by the fact that these studies were conducted under aerobic conditions where yeast cells, as I've just described, are particularly unsaturated, which could perhaps mask the defects associated with unremodeled cardiolipin. And indeed, so we wanted to apply how our microaerobic model, our increasingly saturated model, to better understanding cardiolipin remodeling in these conditions. And so we first looked at strains with Barth syndrome-like mutations in the conserved regions of TAS1, which were generously provided by Steve Claypool's lab at JHMI. And indeed, in aerobic conditions, we saw in these strains a retention of cristae tubule formation. However, specifically under microaerobic conditions, we see an ablation completely of cristae structure, which was also observed in knockouts of TAS1. Indeed, lipidomic analysis of these cells in aerobic or microaerobic conditions revealed that TAS1 knockouts retained a high level of unsaturation in cardiolipin molecules in aerobic conditions. But in microaerobic conditions, we see a significant increase in the saturation of cardiolipin molecules, which could be driving the defects specifically observed under microaerobic conditions. Indeed, we were even more surprised that loss of cardiolipin remodeling initiation by CLD1, which is a cardiolipin deacylase, resulted in more major phenotypes under microaerobic conditions, like a complete loss of cristae structure and an inability to grow on respiratory media after pre-incubation in microaerobic conditions. A phenomenon that was actually specific to increasing saturation, as when we fed cells with unsaturated fatty acids, oleic acid, this completely rescued this loss of this phenotype under microaerobic conditions. Indeed, we wanted to further apply this understanding or the specific role of deacylation in this process to mammalian cells, where in HEC cells, we actually conducted what could be the closest analogous experiment, whereby we inhibited IPLA2 enzymes through bromoenol lactone. And IPLA2 enzymes in mammalian cells conduct an identical reaction to CLD1. And indeed, bromoenol lactone was sort of posited as a previously posited as a therapeutic for Barth syndrome because it limited the formation of this monolysocardiolipin molecule, which was particularly pathogenic within Barth syndrome. Indeed, when we treated cells, these are HEC cells, with bromoenol lactone, we only saw mitochondrial dysfunction specifically when we fed cell or co-fed cells with palmitic acid and not oleic acid, both in terms of defects in respiratory capacity as well as cristae ultrastructure. And indeed, we've also observed a similar relationship in other cell lines. For example, in C2C12 myoblasts, which were generously provided by Miriam Greenberg. We see that specifically the co-treatment of BEL and palmitic acid results in mitochondrial dysfunction that mimics the dysfunction induced by knockouts to TAS when co-treated with palmitic acid. So there's a sort of similar dysfunction observed in BEL plus palmitic acid cells as in TAS-KO cells. Indeed, we sort of integrated these results and derived an understanding of how cardiolipin remodeling may be dispensable or functionally identical under unsaturated lipidomes, but specifically is required at least to shape mitochondrial structure and function under saturated lipidomes. And we're currently investigating a new enzyme that we've characterized involved as an which could be an alternative pathway involved in cardiolipin remodeling, known as DDL1, which is also a PLA1 similar to CLD1. And we found that its knockout increases cardiolipin levels in both unremodeled and remodeled remodeled and unremodeled backgrounds, and also rescues uh, defects to cardiolipin remodeling under conditions of increased saturation. So we're currently investigating the roles of DDL1 in cardiolipin remodeling, and also the functions of its human ortholog DDHD1 and its potential inhibition in mammalian cells, and whether this could increase cardiolipin levels in these tafazin mutants. So in summary, I hope I've delineated how lipids could be influencing the structure and function of mitochondria, and particularly its physical properties, and also whether how this model could be applied to understanding the cell type heterogeneity that's been observed in Barth syndrome, as cardiolipin remodeling could be a significant function of the surrounding lipid environment. And with that, I'd like to acknowledge my PI, Itai, who's been a fantastic mentor, as well as the rest of my lab, and significant and fantastic collaborations with the Ellisman lab, the Lippincott-Schwartz lab, who provided electron microscopy assistance, the Rangamani lab, and the Lee lab, who were instrumental for the molecular dynamics simulations we conducted. And also that these titans in the cardiolipin remodeling field, the Claypool lab, Gohill lab, and the Greenberg lab, who not only provided reagents, but also helped us rationalize some of the results that we were observing. And with that, I'll take any questions. Thank you.
All right. Thank you, Kailash, for a fantastic talk. Just a reminder to everyone to raise your hand and can call on you for questions. So while we wait for those to trickle in, I was going to ask you, have you looked at all at recovery from your different treatments to kind of watch the reverse to see if there's recovery to the mitochondrial morphology and cristae, or to kind of examine how modeling works in the opposite direction?
You mean recovery as in, yeah, so normally we see recovery if we do an analogous treatment in unsaturated conditions. So supplementing cells or saturated cells with oleic acid, even the genetically modified ones, completely recover the cristae structure.
Right. I guess I'm wondering about the dynamics. So have you ever tried going and watching kind of a time course of,
Mhm.
from microaerobic back to aerobic, particularly in like your cardiolipin synthase mutant?
Oh, that's a really interesting question. Yeah, we haven't actually observed, we haven't looked directly at that, but that's something definitely that we should look at. Yeah.
Okay. I'm not seeing any other hand raise, unless I'm looking in the wrong place. Oh, there we go. We have one from Vishal Gohil.
Yes, can you hear me?
Yes.
Kailash,
Hi.
very nice talk. Good to see such wonderful stories coming from you and from Budin Lab. I have a more general question, and so the whole story really is inspired by the idea that in the wild, in the natural environment, yeast Saccharomyces cerevisiae would be exposed to lower oxygen than in the aerated flask that we grow in lab.
Mhm.
So, I mean, I'm so a bit confused about that because the yeast are growing on the surfaces of fruit. They should have atmospheric oxygen, same 21%. Why do you think that the natural environment is oxygen deprived?
Well, I
Maybe.
guess relative to, I think the shaking and combined shaking and aeration actually is uh, artificially more aerated than when you're not shaking. So I think we'll actually observe even similar phenotypes if we just leave yeast growing on the bench top rather than shaking them at 200 RPM. That also, I think, modifies the oxygen availability as well.
Okay. And is there some data on that you know basically support what you just said right now?
Um, Not that I need to look more into that, but I think, I mean, even the way we grow them in these microaerobic conditions, we specifically see um, changes in unsaturation, but also when we measured, I guess, oxygen levels in yeast cultures just grown regularly, it is significantly lower than when shaken.
Okay, thank you.
Itai has his hand raised, so I'll go ahead. Got it.
Yeah, I was just gonna add to that. I think when things are growing on a surface, like even on a plate, they're in a 3D structure that limits gas diffusion. And so I think, to your question about, growing on a fruit, Vishal, I think that's relevant. And then I think in growing in liquid, as Kailash said, if you grow cells, even how they grow them at in a fermenter at a brewery, to make beer, it's really microaerobic. And that seems to be kind of how, these yeast that we use in the lab sort of evolved.
We also have a question from the chat box that I'll read out to you. So does cardiolipin have increased saturation under either artificial and microaerobic conditions, or is that just for the other phospholipids? Does it look different when you don't knock out tafazin? Basically, I'm curious how if unsaturation in cardiolipin or the head group to acyl chain ratio shapes its structure and function.
Yeah, so under these microaerobic conditions, just in wild type cells, we actually see a specific increase in saturation under cardiolipin. So I guess all of the phospholipids do increase their saturation, but the magnitude of the increase in saturation is largest within cardiolipin compared to other phospholipids. And to answer the second part of the question, let's see how unsaturation in the head group acyl chain ratio affects the structure. Yeah, so we think I guess the actual physical property of cardiolipin in terms of their curvature and the potential, curvature localization could be dependent on the unsaturation itself. When the larger the magnitude of unsaturation on cardiolipin, the more chance that it can localized to regions of higher curvature in cristae itself.
Okay, we've got a question from Steve Claypool.
I can't hear it. I couldn't hear any of that.
Yeah, Steve, I don't know what's going on with your audio, but maybe you want to type your question in. Okay. Thoughts on P to PC changes in mitos versus cell?
In what regard, I guess?
Maybe what we can do is have Steve ask his question and then we can answer it after the second talk.
Yeah, sounds good.
Or you guys can talk.
Yeah.
All right. So why don't we go ahead and switch? Thank you, Kailash, for a fantastic talk. And now we're going to switch gears here, and I'm going to introduce Jason Casler. So Jason is currently a postdoc in Laura Lackner's lab at Northwestern. He did his PhD in Ben Glick's lab looking at Golgi trafficking, and now he's switched gears to mitochondria and has been studying a three-way interorganelle contact between the ER, mitochondria, and the plasma membrane. And he's going to talk to us about some exciting work on how it not only influences mitochondrial organization, but also PI4P distribution at the plasma membrane. With that, take it away, Jason.
Okay, thank you for that. I will share my screen. Okay. So yeah, thank you very much for the opportunity to share my work. This is a really cool venue. So as Jonathan said, I'm a postdoc in Laura Lackner's lab at Northwestern, and I'll be talking about organelle contacts today and how they influence organelle function and lipid distribution. So biology has long progressed past the idea of organelles as these sort of static, independent structures, and we now recognize that they form these really dynamic, interconnected networks, as illustrated by this beautiful movie from the Lippincott-Schwartz lab, where they simultaneously labeled several different organelles. You can see them bumping into each other. There's all kinds of interconnectedness in these networks. And one of the ways that cells have evolved to organize these networks is the formation of what's known as membrane contact sites. So these are sites of close apposition between different organelles that don't result in full fusion. And here are just a few really beautiful examples you can see from electron microscopy data. You can see these large stretches where the two organelles are in close proximity. And these are actually highly regulated areas where organelles are communicating and exchanging metabolites. So our lab is primarily interested in understanding mitochondria and the functions of the mitochondrial network and how they're regulated. And it turns out that membrane contact sites are a very critical part of this network. From this cartoon that Laura made a few years ago, she was just summarizing the different kinds of organelle contact sites that we knew about that mitochondria make with other organelles. And the short answer is they seem to contact everything and be involved in tons of different functions. So some of the overarching questions that I've been interested in are trying to identify some of the actual molecules that are involved in tethering. While we can see these things very clearly from electron microscopy, we don't always have a very strong grasp on what molecules are involved. And then importantly, what are the actual functions? And something that I've been really interested in is how does the actual organelle tethering function contribute to these various functions? And so since these are really broad questions that are difficult to answer, we've decided to focus in on a model contact site and try to dissect it in really exquisite molecular detail to try and understand some of these broader principles. And the one I'll be talking about today is known as MECA for the Mitochondria-ER cortex anchor. So this is actually a tripartite membrane contact site between three different organelles, the plasma membrane, the ER, and mitochondria. It consists of at least three different protein components. Num1 here is the central tethering molecule that I'll be talking a lot about. MDM36 is thought to help cluster or oligomerize Num1 into these higher order structures. And then most of the talk will be centered around a recent identification of the ER interacting partner, SCS2. And I should mention that all of this work is taking place in the simple budding yeast, Saccharomyces cerevisiae. And so our group and others have been working on this contact site for some time at this point, and we have a pretty detailed molecular picture of how it's interacting with its different molecular components. So Num1 is unique in the fact that it has two lipid binding domains. It has a pH domain that interacts with PI45P2 on the plasma membrane, and it has a coil-coil domain that interacts with lipids on the mitochondrial outer membrane. And then we've characterized most recently this interaction between SCS2 and a fat motif in the C-terminus that I'll talk about in more detail soon. So if we look at this in actual cells, here's what it looks like. Here I've labeled two different components of MECA, Num1 in blue and MDM36 in green. And it forms these cortical clusters that are tethering the mitochondrial network over time. And up here in the top, you can see a really clear example where the mitochondria is sort of tethered to that specific point throughout the course of this 10-minute movie. So what are some of the functions of this contact site? The most visibly obvious one is that it controls the positioning of the mitochondrial network. In wild-type cells, mitochondria are positioned cortically, distributed around the cell, whereas in a Num1 deletion, they collapse into the center of the cell. This is also involved in regulating mitochondrial fitness, though I won't have time to discuss all of the data for this, but these cells on the left were grown in fermentation conditions. On the right, they're grown in respiration conditions, and you can see a drastic upregulation of the mitochondrial network and a, oops, sorry about that. You can see a drastic upregulation of the mitochondrial network that is not as robust in the Num1 deletion. And so since we've been working on this for some time, we again have characterized many different functions for this contact site. And today I'll be particularly talking about how the ER tethering function of this contact site is involved in regulating mitochondrial division and a novel connection we made to PI4P metabolism. Before I go any further, I wanted to thank three former graduate students in the lab who have all gone on to excellent careers who helped make this work possible. Claire, Antonine, and Heidi all contributed significantly, and I'll try to mention them when they come up. So for the first part of the talk, I'll talk about mitochondrial division. For those who are less familiar with this organelle, mitochondria form these elaborate networks that are constantly being remodeled through fusion and fission events, as shown here by this blue arrow. And these are really important to maintaining the metabolic and genomic health of mitochondria. Now, it had been known for some time that loss of this tethering complex causes a change in mitochondrial division, but it really wasn't that carefully understood or carefully dissected. And so Claire, when she was a graduate student, went and just counted the number of mitochondrial division events you can see over time. And you get about a 50% reduction when you lose NUM1, which is not as severe as full loss of the mitochondrial division machinery. And one of the questions we wanted to ask was, what is the actual mechanism by which this is happening? So we had several clues to how this might be occurring. So our host, Jonathan Friedman, when he was a graduate student in Guy Vogt's lab, collaborated with my current boss, Laura, when she was a postdoc in the Nunari lab to publish a really excellent paper where they demonstrated that these Mitochondria-ER membrane contact sites are critical regulators of mitochondrial division. And as you can see in these images, these mitochondrial division events are always coupled with the presence of ER. And so prior to joining the lab, we didn't actually have an entire molecular mechanism for how NUM1 was interacting with the ER. Through some work, I don't have time to get into all the background, but we identified this FAT motif or F-FAT motif in the C terminus of NUM1 that is sufficient to drive localization to the ER. And this stands for 2-phenylalanines in an acidic tract. And these are well-characterized motifs that interact with what in yeast is known as SES2. In mammals, this is known as the VAP proteins. And these are well-characterized to recruit different classes of proteins to the ER that contain fat motifs. So just to verify that these were actually interacting in vivo, we did biochemistry. If you delete the fat motif or just mutate a key residue, you can completely block co-immunoprecipitation. And if you lose that fat motif, you reduce the co-localization in cells as well. So these are definitely interacting in vivo. And what was exciting about this to us was it allowed us to generate a separation of function allele that no longer functioned as a mitochondria-ER plasma membrane tether, but only as a mitochondrial plasma membrane tether. And so on the left, you're looking at full-length NUM1, and it's the organelles it's tethering. All of the blue foci are hard to see because they're covered by both mitochondria and ER. On the right, I've deleted the fat motif, and it's still functioning as a mitochondrial plasma membrane tether, but now that green ER is not present at those sites anymore. So using this allele, we wanted to ask whether or not this could explain NUM1's role in regulating mitochondrial division. And the answer was yes. Simply deleting the fat motif was sufficient to completely recapitulate the full loss of this tripartite membrane contact site. And so this was exciting to us because even though these have normal morphology, they're no longer showing wild-type division rates. So we were able to pretty strongly pinpoint a function to a specific tethering function of MACA. So, and again, this is exciting to us because a lot of membrane contact site research tends to stall at the idea of putting actual mechanistic details into how these contact sites are working. And so now I'm going to switch gears a little bit and talk about a novel function that we identified. And then at the end of the talk, I'll come back and discuss how I think these two are actually related. And so this was our discovery that this contact site is also involved in regulating PI4P metabolism. So like I mentioned, we were very interested in understanding all of the different functions of this contact site. And to try and identify novel interactors, Antonine performed an IP mass spec experiment. And we got all of the protein components we were hoping to see, all the components of MECA come down, some of the mitochondrial division machinery, which was already known to interact with MECA. But she also saw something interesting. We also had weak enrichment of every component of a plasma membrane localized kinase machinery that's involved in synthesizing PI4P. And this was very interesting to us, even though the hits weren't super well enriched, because there's a lot of interesting connections between these kinase patches and MECA. So we decided to look further into this. These are known as PIC patches or phosphoinositide kinase patches, and they're involved in the synthesis of PI4P, phosphatidylinositol 4-phosphate, from phosphatidylinositol, specifically at the plasma membrane. So STT4 is the kinase that is actually phosphorylating the lipid. YPP1 and EFR3 are scaffolding proteins. And interestingly, these were already known to interact with SES2, that same ER interacting or ER recruiting protein we identified as a component of MECA. And so these are well known to localize to plasma membrane ER contact sites, where they're involved in the transport of PI4P back to the ER, where it's dephosphorylated by the phosphatase SAC1. And I don't have time to get into all of the literature, but there's a really rich body of literature about how this mechanism is involved in the countertransport of different lipid species at membrane contact sites. So all of this was really interesting to us, and we decided to look at if there was any functional crosstalk between these two machineries. And so by fluorescence microscopy, these things seem to co-localize. It's not an exact pixel for pixel overlap, but more of an adjacency. And by co-IP analysis, we also detect interaction as well. So even though our IP mass spec wasn't the strongest interaction, these definitely seem to be interacting in vivo. And so the next thing we wanted to do is see if there was any functional crosstalk between these. And so we decided to look at the output of these kinase patches, which is the formation of PI4P. And I'm a microscopist, so I decided to look at a PI4P biosensor. So this actually has a really beautiful localization pattern. In wild type yeast, you see a strong enrichment on the plasma membrane of the growing bud, a very weak enrichment on the mother cell, and these intracellular punctate spots are actually synthesized by another PI4 kinase known as PIC1, which is not the focus of the talk today. So if you mess with this reaction, if you get rid of SAC1, you drive the reaction towards the synthesis of PI4P, and you see a drastic change in the localization of the biosensor, because now there's so much PI4P on the plasma membrane, the biosensor is completely relocalized. The Golgi pool still exists, it's just in lower concentration. Now, when we deleted num1, we saw something very interesting. Instead of that normal enrichment of PI4P on the bud, we now saw an accumulation of PI4P on the mother cell plasma membrane as well. And this is indicative of something going wrong in this standard metabolic pathway. So as I mentioned, I'm interested in understanding how contact sites are actually regulating these different types of functions. So I wanted a way to look at if we could tie this to any of num1's organelle tethering functions. So I devised a method to quantify the enrichment of PI4P on the plasma membrane of the bud compared to the mother, just by plotting the ratio of the two fluorescence intensities. And you can see in wild type cells, we get about a 5, almost 6-fold enrichment, whereas when you delete num1, that reduces significantly. And by using a host of different mutants, including that num1 delta fat allele I mentioned earlier, we were able to ascertain that actually all of its organelle tethering functions are at least partially involved in regulating this function. So this is one of the first examples of a true function of the tripartite membrane contact site that we had. So I'm interested in mechanism and understanding how these things are actually working, and we wanted to ascertain how we're actually getting an accumulation of PI4P on the plasma membrane in the mother cell. And the most obvious candidate was that we were somehow disrupting the turnover of PI4P from the plasma membrane to the ER. And this is due to the fact that these are actually fairly well-studied processes, and other groups have identified similar mechanisms before. So I wanted a method if I could actually measure the rate of turnover. And to do so, I devised a strategy to inactivate pick patches. These are essential protein complexes, so you can't delete them. I used the auxin-inducible degradation system to conditionally deplete a component of pick patches, which results in the loss of PI4P from the plasma membrane. And now you only see the Golgi pool after adding auxin. And so what we want to use this system for is to inactivate the SDT4 plasma membrane kinase and then measure how quickly we're losing the PI4P pool as it gets dephosphorylated back into phosphatidylinositol. And this should be a way to indirectly measure this rate of turnover. And so here's an example of what it looks like in wild type cells. If you add just vehicle control, you maintain PI4P on the plasma membrane. If you add auxin, you start to see a rapid loss of PI4P. And within about 20 minutes, you've already lost the majority of it. And so when we added our num1 deletion into this system, we saw something really interesting. Even after that same time point of 20 minutes, cells still have a significant amount of PI4P on the plasma membrane. And it does go away eventually if you take these quantifications out longer. Here, the black line is showing you the num1 deletion in comparison to the wild type, this blue line. It does deplete, but at a much, much lower rate. So what we think is happening is that when you lose MECA, you're significantly disrupting the ability of the cell to turn over PI4P by transporting it back to the ER for dephosphorylation by SAC1. And this likely has broad effects on several different lipid trafficking pathways, although we haven't actually measured any of them, because this PI4P countertransport pathway is well known to be involved in lipid trafficking. And one of the exciting things about this is it implicates a novel membrane in this system that previously was not known to be involved. And so we speculate, and we're currently involved in following up with experiments to see how much of a role mitochondria actually play in PI4P metabolism. And so then just to summarize the second part of the talk, I demonstrated that the tripartite nature of this mitochondria-ER plasma membrane contact site actually regulates the efficiency of PI4P turnover. Now, I mentioned I was going to circle back and discuss whether or not these phenotypes were related. And I think they are, and I think there's a lot of interesting work that's been coming out that shows that PI4P might play a much larger role in mitochondrial biology than we had previously thought. So here, I'm just going to go through some of the data, the interesting data implicating PI4P in the regulation of mitochondrial division. So the whole story I just told you was about a tripartite contact site and the plasma membrane pool of PI4P, and how this contact site is also involved in the regulation of mitochondrial division. Work from the Prudent lab and Heidi McBride demonstrated that post-Golgi TGN vesicles that are enriched in PI4P are actually present at sites of mitochondrial division as well. In addition, lysosomes, which contain PI4P, have been shown to be involved in mitochondrial division. And this one actually has some implication that a specific transport protein, ORP1L, is involved in this process. And then finally, there are proteomic data that indicate that PI4 kinases are actually present on the mitochondrial outer membrane. And I'm not including it here, but there are some really nice studies from the Hammond and Bala labs that demonstrate that mitochondria actually have a relatively large pool of phosphatidylinositol, which is fairly interesting because that could be either a precursor to the synthesis of PI4P or the dephosphorylation product of PI4P. And so this is all just sort of hand-wavy speculative ideas, but I think it's a really interesting a set of observations that merits significant further investigation. And then finally, just to conclude, I wanted to bring back sort of what we think this sort of multifunctional contact site is doing in yeast. And I think what it is a primary example of how these membrane contact sites can serve as organizing hubs that can regulate the efficiency of different biological processes. So MECA is pretty unique in that it's very, very stable, and that cells can use it as a platform to organize different transport pathways between multiple membranes. And with that, I would just like to thank everyone in the lab. Laura has been an excellent mentor. Claire, Heidi, and Antonine all contributed to these projects. Funding from the NIH. I was lucky enough to get an F-32. I hope other people also have that opportunity in the future. And thank you everyone for your attention, and Jonathan for inviting me.
All right, thank you, Jason. It was a great talk. Again, people raise your hands if you want to ask questions. I'll get us started. It's really interesting that you're seeing that in the NUM1 mutation, you're getting a reduction in division rates, but not a complete loss. And I guess putting the two stories together, do you think that the mitochondria near the PM are playing, like the cortical mitochondria are playing a different role, or that those divisions are different? And kind of related to that, do you think these aspects of mitochondrial differences might extend to mammalian cells in some way?
Yeah, those are excellent questions. So I will say with the division stuff, we didn't actually measure like when you delete NUM1, if you're seeing a reduction in the number of like, we didn't measure spatially inside the cell, like where the loss and division rate is happening. I will say I have actually done these experiments with an ER marker as well. And the vast majority of them, as seen in wild type cells, are still ER associated, which we would have expected. So that being said, I do think there probably is something somewhat unique about the mitochondria that are sort of tethered to the cell cortex. We've done several different types of experiments now where we've sort of increased or added synthetic tethers to add more mitochondria plasma membrane tethering. And you do see reductions or changes in the number of mitochondrial division events. I'm not entirely sure exactly what those would imply. But then in relation to the connection to mammalian cells, in one of those papers I cited earlier, they actually did a double knockdown of VAP-A and VAP-B in mammalian cells and saw a significant reduction in the number of mitochondrial division events, very similar to what we saw in yeast. So I think that there's almost certainly some sort of conservation of at least a VAP-mediated function in mitochondrial division.
We have a question from Anat Manan. Unmute him.
Yeah, hi, Jason. You have a sort of very nice system for, well, your data show that you can disturb PI4P metabolism or turnover in the NUM1 deletion cells. And I was wondering if you have any evidence of the consequences that you advertise, which is that the client lipids in these counter transport pathways, are they dysregulated? So is PS, for example, no longer distributed primarily to the cytoplasmic face of the plasma membrane? Is ergosterol not concentrated in the same way at the plasma membrane, and so on?
Yeah, thank you. That's a really excellent question. So I personally have only looked at I've looked at a few different biosensors. So I've looked at a PS biosensor via fluorescence microscopy, and I didn't really see any obvious changes. We haven't done any sort of careful lipidomics, especially not targeted to specific organelles or anything like that. So if there are changes, they're subtle enough to not see them by fluorescence microscopy with biosensors, but I have not done a whole rigorous characterization of the many different pathways that are known to be involved with PI4P countertransport. It's definitely something we're interested in looking at.
So the reason I asked is because this allows you to perhaps assign a weight to these counter transport pathways versus other ways to get these lipids between the endoplasmic reticulum and the plasma membrane. So the counter transport is very, seductive, attractive, sounds very nice and so forth, but I don't know how much weight it has in terms of overall lipid trafficking in an unperturbed cell. And so you have a means to do that. You have a means to measure it.
No, that's absolutely a great point. And I think that is a key next set of experiments, is that we're going to look at other types of lipids and different lipid transporters.
Thank you.
Okay, we have a question from Kentaro Sakata.
Yeah, hi, yeah. I'm Kentaro Sakata. I'm also postdoc in the Melon Lab, so I'm just confirmed about the. Do you ever check about the PIK1? The PIK1 deletion is also give the same phenotype as the STT4 knockdown.
So if I understand the question correctly, you're asking about PIK1, which is the Golgi localized PI4 kinase. I personally haven't worked with that mutant. We have temperature sensitive versions of PIK1. It's also essential, so we can't delete it or anything like that. We haven't looked at it very strongly, but it's something that we're interested in. What I will say is that the two PI4 kinases are at least thought to be somewhat independent of one another, because if you get rid of STT4, the Golgi PI4P pool is still there. If you get rid of PIK1, Scott Emmer's lab showed that the plasma membrane pool is still there. So they at least operate independently in that function. Of course, there's almost certainly going to be some crosstalk, and I think it'd be a really fascinating research direction to look at if the Golgi pool in particular in yeast is having any effect with mitochondria, because I suspect there's something interesting going on there.
Oh, okay. Yeah, thanks. Yeah, that's what I want to know about. Yeah, thanks. Great.
Well, it doesn't look like any more questions are popping up in the chat. So I just want to take one more opportunity to thank you, Jason, and also Kailash for giving fantastic talks. It's been a great session, and thank you all for attending as well.